everyone, welcome to my channel Srinivasa Biology Tutorials. In this section to discuss NCERT class 12th, the sexual reproduction in flowering plants, part 6, that is double fertilization. Double fertilization. After the entering one of the ncert, the pollen tube release as in two male gametes into as in a cytoplasm of the cnidid cells. So here once when you observe to as in a here to get released as in the pollen grains here. So when the pollen grains to get released, what happen here to get pollen tube will be formed in this pollen tube to get forms like, am I right? And here in this enter the pollen grain to get released as in a two male gametes. To release as in a two male gametes, it is in a the first male gamete. It is what? It is in a that is second male gamete. First male gamete and second male gamete to release to as in the in that region has an cnidid cells as present here. Next, one of the male gamete moves towards the egg cell and fuse with its nucleus. So one enter the male gamete, they fuse with as an egg cell of the nucleus. So when the first male gamete and then egg cell they get fused, this total process is called as an syngamy. This process is called as what? The syngamy process. This result the formation of the diploid zygote. So here the first male gamete is in haploid condition, egg cell also has in haploid condition. So haploid condition to form as in a diploid zygote is formed. Next, here the other male gamete to move towards the polar nucleus located on the central cell and the fuse with them the produced as in the triploid primary endosperm nucleus, that is pen. So here they having as in completely the polar nucleus is present or nothing but as in a central cell. The second male gamete they fuse with as in a here. So it is in a polar nucleus or central cell, they have two in, it is a one nucleus, it is a one nucleus, the two nucleus, and here the second male gamete. So these three are to get fused, the two cells and here one cell, the total three cells are to completely get fused here. When to get formation of what? Pen, that is in a primary endosperm nucleus is produced. This involves the fusion of the three haploid nucleus. It is the termed as a triple fusion. So here how many cells are fusing here? Three cells are fusing. These are the central cells or the two cells, and here male gamete is in a one cell. The total how many? Three cells as they are fused. So hence it is called as what? That is in a triple fusion. Since the two types of fusions of syngamy, the triple fusion, it takes place in the embryo sac, the phenomenon is double fertilization. So here the first the syngamy will be going on, as well as in a here triple fusion also they going on. The here two times here completely the fertilization will be going on. So hence it is called as what? Double fertilization. It is called as what? Double fertilization. Next, an event unique to as a flowering plant. So when you observe as in the flowering plants, most this kind of event will be there takes place. Okay, and here the central cell after the triple fusion becomes as in a primary endosperm cell. So here the primary PEC means primary endosperm cell. Primary endosperm cell to get formed here. That after that develops into as what? Endosperm, while the zygote develops from as an embryo. So the here the primary endosperm cells convert as in the endosperm and zygote is developed from as what? That is in a embryo, developed as in a embryo. Let us see here the diagram. Let us see that in the diagram. See the diagram. See, these are the completely in degenerating cnidid cells and zygote and primary endosperm, that is in a completely the PEC, and it is in a primary endosperm nucleus, that is in a pen. So after it is the development of this enter the embryo. Let us here what are the post fertilization events? What is that in a post fertilization events? Let us see. The following the double fertilization events of the endosperm and embryosome development, maturation of the ovules into seeds and ovary to make as any fruit, or the collectively termed as in the post fertilization events. So after the double fertilization, here endosperm and embryo is developed as any ovules into the seeds and ovary make as any the fruit or the collected the term known as what? That is in a completely post fertilization events. Let us to discuss endosperm. Endosperm. The endosperm development, the precedes embryo development. Why? The primary endosperm cell divided is as in the repeatedly and form as in a their triploid endosperm tissue is formed. Here primary endosperm tissue is formed. The cells of this tissue are filled with as in the reserve food materials and they're used for as in nutrition of the development of the embryo. So inside of the enter the embryo sac they having as what? Inside of the embryo sac here they filled with as in total the nutritions as present, that nutritions they get used as in a this completely embryo sac. Next, after in this most common type of endosperm development, the pen undergoes successive nuclear divisions to give rise to as in a free nuclei. It is further the development to get formed as what? To make as in that free nuclei. This stage of the endosperm development is called as in a free nuclear endosperm. So when to get formation of the free nuclear, that stage is called as what? That is in a free the nuclear endosperm. So subsequently, the cell wall, the formation of occurs and the endosperm becomes the cellular. So the enter the cell wall is developed to as what? It is a part of the cellular endosperm. And the number of the free nuclei formed before the cellularization varies greatly. When you observe to as in that before the development, we get formed as in the cellularization of the free nucleus is formed. See that in a coconut water from as a tender coconut that you are familiarized with is the in nothing but the free nuclear endosperm. They are made up of the thousands of nucleus and the surrounding was in that white their kernels is the cellular endosperm. When you see that in a coconut water, that is nothing but as what? It is a part of the completely free nuclei, and that enter the free nucleus they covered with as in that entire the white color their kernel. It is also part of the cellular endosperm. Okay, and let's next here the endosperm either the completely consumed by the development of the embryo. Endosperm either they're completely they're consuming by the development of embryo, like when you observe as in pea plants, groundnuts, beans. They are before the seed maturation or it may the persistent in the mature seeds when you observe as in a castor and then coconut. Castor and coconut, and these used up during the seed germination. The splits open in some seeds like a castor, peas, beans, groundnuts. The fruits of the coconut look for the endosperm of each case. The find out the whatever the endosperm they persistent in cereals, wheat, rice, and maize. So in that seeds they complete the endosperm they are persistent because the enter the seed coat as well as endosperm they can complete their fuse for that in a rice, wheat, and then maize seeds. Okay, yeah, like next when you observe to as an embryo, when you see that in a one particular the embryo structure, see it is a completely embryo structure here. See the embryo develop at the micropolar end of the embryo sac where the zygote is situated. So mostly the embryo is developed as in the micropolar end where the zygote is situated. The most of the zygote is divided after the certain amount of the endosperm is formed. The zygote is developed. It is certain amount of only for endosperm. Before only we discuss what that enter the embryo to get utilized as an endosperm, then they get developed, right? Like this is in the adaptation to as in provide the assumed nutrition to as a development or developing embryo. It is to complete it to development of the embryo only. Through the seeds differ gender greatly and the early stages of embryo development that is called as an embryogeny. So early development of the embryo that is called as what? Embryogeny. Next, the are in similar the both of the monocotyledons and dicotyledons. When you observe to as in that monocot the embryo is different as well as in a dicotyledon embryo is they are different, and they given as the figure also in 1.3 that depicts the stage of the embryogeny in dicotyledon embryos. Here the zygote it gives rise to the in a pro embryo and then subsequently to the in a globular heart shape and mature embryo. The previous diagram we see in that they appear as in a globular heart shape and their mature embryos are we noticed. See, when you observe here most of this diagram here, this is a complete development of the in a heart shaped embryos are noticed. A typical dicotyledon embryo, it consists of as an embryonal axis and two cotyledons. Basically, when you observe to as an one particular the dicotyledon seed, it is to get composed of as an that embryonal axis and to get form as in a two cotyledons. See, these are two cotyledons. It is in a embryonal axis. This is in a embryonal axis. These are in a cotyledons. Cotyledons. The portion of the embryonal axis above the level of cotyledon, it is an epicotyle, which is terminates with as an plumule or in a stem tip. In the cylindrical portion below that in a that is called as a colio, the cotyledon that is in a hypocotyle. See, it is in a hypocotyle, that is in a epicotyle, and that the terminates to as in a lower end the radicle or root tip. The root is covered with as a root cap also present. See, the root cap, radicle, and these are completely coleorhiza. And the embryos of the monocotyledons possess only one cotyledon is present. When you observe as in monocotyledons, here monocotyledon possess as what? In only for single cotyledon is present. Like as in a grass family, cotyledon is called as in a scutellum. In that grass, it is in a dome shape. Here, the dome shape or shield shape. Here the shield shape structure is present, that is called as what? Scutellum. This is situated as towards one side lateral to the embryonal axis. It is a lower end, the embryonal axis has the radicle and the root cap enclosed in the undifferentiated sheath is called as an coleorhiza. That is called as what? Coleorhiza. And the proportion of the embryonal axis above the level of the attachment of the scutellum is in epicotyle. So epicotyle means upper side, that is a here. Epicotyle means it is a form of the scutellum above the region, and the epicotyle has in that shoot apex and the few leaf primordia enclosed halo the follicular structure is called as an coleoptile. So it is the form of the cap-like structure you noticed as in the shoot apical meristems. That is an in that coleoptile. And the soak a few the seeds in water, say of water, wheat, maize, peas and chickpeas, groundnuts, the overnight, then the split the seeds and observe the various parts of the endosperm, the various parts of the embryo and the seeds. So when to get observe what happened to get development is takes place. Okay. Next, after is what? Next to seeds. Next one is what? Seeds. Here, that angiospermic. In a angiosperm, the seeds it is in the final product of the sexual reproduction. Seeds are nothing but as what? It is in a final the production of the angiosperms. See, it is often described the fertilized ovule. It is described as a fertilized ovule to get formed as seeds. Seeds are formed inside the fruits. A typical seeds consist of as seed coat, cotyledons and embryonal axis. Before we discuss that one only, they are having a seed coat as well as an embryonal axis and then complete cotyledons as present. The cotyledons, the embryo is a simple structure, generally thick, swollen due to the storage of food, reserved food materials, legumes, like we observed as a mature, maybe the non-albuminous or exoalbuminous seeds. Here, that enter the generally they are thick, swollen due to the storage of food reserved materials, right? They stored as a food materials that is called as a non-albuminous or exoalbuminous. Non-albuminous seeds have the residual endosperm that completely they are consumed during the embryo development. So when the development of the embryo to complete utilized as the total endosperm. Example say as pea plants and groundnuts. And albuminous seeds that retain the part of the endosperm, it is not completely utilized during the embryo development. Example, wheat, maize, barley, castor. So in this species, that entire the endosperm is not utilized for the development purpose. Occasionally in some seeds such as in black pepper and beet, the remains of the nucellus here are the also persistent. So we can't differentiate it as in the total in the endosperm like. So that is persistent condition. This residual, the residuals present at the nucellus is called as a perisperm. That is called as what? Perisperm. Okay. Next, after the integuments, here the integuments of the ovules, the hardened touch the protective seed coats. So here integuments get act as what? They are the protective layers. Next, the micropyle remains, the small pore is present, that is called as a seed coat. The familiar, the facilitated entry of the oxygen, water into as in a seed during the germination. So sometimes when the seed is germinated, they require as in a nutrients and air, water, gases is required, oxygen, carbon dioxide, whatever. So the total they get a transport with help of the small pore. Okay, the seed matured, its water content reduced, and the seed becomes the relatively dry up to as in a 10 to 15% the moisture by mass. So when they get mostly the reduced, how much the seed approximately dry into as in a 10 to 15%. Generally the metabolic activity of the embryo slow down. So in that condition, what happens? Metabolic activity there gets slow down. The embryo may be there enter the state inactively, that is called as a dormancy. So the time what happened, nutrition is not available and temperature is not perfect, then what happens? Seed go to as in the inactive condition. That condition is called as what? Dormancy condition. And or you give here give it as another definition also. Or if the unfavorable conditions are available, adequate the moisture, oxygen and suitable temperatures, they get germinates. So when favorable conditions comes, what happen? It is again to get germinates. Okay. Next, ovule, as ovules mature into as a seeds, the ovary develop into as a fruit. The transformation of ovules into the seeds and ovary become as a fruits produced as simultaneously. So here already we discussed what here ovaries become as a fruit and ovules become as a part of the seeds. Okay. Next, the wall of the ovary developed into as a wall of the fruit called as pericarp. So already you know that when you take as any one particular fruit, in this fruit which has any three parts is present. Am I right? What is in epicarp, outermost layer, and this middle edible part is called as a mesocarp, and then last stony part, that is in endocarp. So in these three layers we call as what? That is called as in a pericarp. These three layers fused is called as what? That is in a pericarp. Now the fruits may be the fleshy, like as in a guava, orange, mango, etc. So this fruit is when ripening time it is fleshy, that is called as a fleshy fruits. Example like as a guava, orange and mango. In or maybe the dry, when the ripening time the pericarp is dry, that is called as a dry fruits, right? Like as a groundnuts, mustard, etc. Many the fruits has uncovered as their mechanisms for the dispersal of the seeds. So when inside of the name mature that enter the seed to dispersal to and dehiscence of the seeds. The recall your classification of the fruits and their dispersal of the mechanism that you have studied about as in the earlier class. So the already in the morphology chapter we discussed what is in the dispersal of the pollen that seeds. Okay, dehiscence, dry dehiscence, dry indehiscence, and caryopsis, and cypsella, and sorosis. These are already completely we discussed in that in a last section. Okay, and these are the relationship between as a number of the ovules in and the ovary and the in a number of the seeds the present on the fruit. The number of seeds, the number of fruits as present on the particular region. The remaining to discuss in next class. Thank you for watching.